Welcome to the deep dive. Okay, imagine trying to sort this tangled mess of threads, right?
Mm-hmm.
Different sizes, static cling everywhere,
Yeah.
and you just want to sort them by length.
Yeah, that was basically the challenge for early protein scientists.
Exactly. And then, boom, this technique comes along. SDS-PAGE. It basically straightens them out and sizes them up.
It really did change the game.
So today, we're diving deep into this really essential lab technique. It's like a shortcut for scientists to separate and study proteins.
Yeah.
You find it everywhere: biochem, molecular biology, biotech. It's fundamental.
Said it's so important why you need that separation. It's not just, oh, how big is it?
Right.
It's about purity, identity, how much is there? I mean, that's the foundation for, well, countless discoveries.
Okay, so let's get into it. SDS-PAGE, sodium dodecyl sulfate polyacrylamide gel electrophoresis. That's a mouthful.
It is, yeah.
But the name kind of gives it away, doesn't it? What's the key part in there?
Absolutely. For me, it's the SDS bit, sodium dodecyl sulfate.
Okay.
That's maybe the unsung hero.
Right.
It's a detergent, right? And it does something brilliant.
Uh-huh.
It unfolds the proteins, makes them linear chains,
Okay.
and it coats them all in a negative charge. Completely swamps their natural charge.
Ah, so they all become sort of uniformly negative?
Exactly. Takes their own charge differences out of the picture. Then the rest kicks in the polyacrylamide gel electrophoresis.
Right, the running part.
You apply an electric current, and because they're all negative, they move towards the positive end.
Okay.
But they have to go through this gel, this polyacrylamide gel matrix.
It's like an obstacle course.
Sort of, yeah, a molecular sieve.
Uh-huh.
And now, since charge doesn't matter anymore,
Uh-huh.
their speed is just down to their size, how easily they can wiggle through that mesh.
So smaller ones zip through faster.
Precisely. They travel further down the gel, the big bulky ones get tangled up more, move slower, stay near the top. Simple as that, really.
It sounds simple, but getting those conditions right must be crucial. What do you actually need, like equipment-wise, chemical-wise, to run one of these?
Okay, yeah. So besides the SDS, obviously you need your protein samples mixed in a special buffer.
Right.
Often has glycerol in it, helps the sample sink nicely into the little wells in the gel, and a tracking dye.
Oh, the blue stuff you see running down.
That's the one. It helps you see how far the run has progressed, so you don't run your small proteins right off the end.
Oh, makes sense.
Then the gel itself, polyacrylamide, the percentage you make it at is key.
Why's that?
Higher percentage means smaller pores, a tighter mesh, good for separating small proteins. Lower percentage, looser mesh, better for bigger ones.
Okay, tunable.
Exactly. You also need a running buffer in the tank to conduct the electrosphere,
Got it.
the electrophoresis box, the power supply, and then afterwards something to actually see the proteins, a stain usually.
Like kumasi blue.
Kumasi blue is the classic, yeah. Stains the protein bands so you can see where they ended up.
So you've done all that. Loaded it, run it, stained it. What does this actually tell you? What are the big applications?
Oh, loads. I mean, the most basic is estimating molecular weight. You run known standards alongside your unknown.
Right, like a ruler.
Kinda, yeah. See how far your protein ran compared to the known sizes? Super useful.
Okay.
Then, checking purity. This is huge. If you're making a protein, say, for a drug,
Mm-hmm.
you need to know it's just that protein, not a whole mess of other stuff. SDS-PAGE shows you if you have one clean band or lots of contaminants.
Critical for safety, I imagine.
Absolutely critical. You can also see how much protein is there, relatively speaking. Like, track if a cell's making more or less of a protein under certain conditions.
So monitoring expression levels.
Yep, and even diagnostics. Sometimes disease states change the protein profile in your blood or tissues,
Ah.
and you might see those changes, like extra bands or missing bands on a gel. It can be an indicator.
Wow. Okay. And it preps for other techniques, too.
Yeah, often it's the first step before something like a Western blot, where you then use an antibody to detect a specific protein within that separated mix.
Sounds powerful, but also like things could go wrong.
Yeah.
You mentioned troubleshooting. What are those common headaches?
Yep.
The smiling bands.
Ah, yeah, the dreaded smile. That often happens if the gel heats unevenly during the run, the edges run faster or slower than the middle.
Okay.
Other big ones, using old reagents. Especially buffers. They can go off, mess up the pH, and ruin your separation. Fresh is best.
Always.
Overloading the wells is another classic. Too much protein just smears down the lane.
Right, just a blob.
Exactly. Or getting no bands at all. Could be a problem with your sample, maybe it degraded, or maybe the staining just didn't work.
Lots to keep track of. And the gel percentage choice, too.
Yeah, choosing the wrong percentage means your proteins of interest might be all bunched up or run too close together to tell apart. You need the right resolution for your size range.
Got it. So wrapping this up a bit, SDS-PAGE, sodium dodecyl sulfate, polyacrylamide, gel electrophoresis.
Yeah.
It really is just a very clever way to sort proteins just by size.
Mm-hmm.
You use SDS to wipe out charge differences and unfold them, then electricity pulls them through that gel sieve.
Pretty much sums it up.
And it gives you crucial info: size, purity, expression levels, helps with diagnostics, and knowing those few troubleshooting tips helps avoid the headaches.
Definitely. And the bigger picture, really, is that even though the name sounds complicated,
Yeah.
it's fundamentally a simple but incredibly powerful tool. It gives researchers this vital window onto the proteins they're studying.
A first look almost.
Often, yeah. It's
Yeah.
very often the starting point before you do more complex analyses on, whatever protein you're interested in, enzymes, antibodies, anything.
It really makes you think.
Mm.
When you consider just how much we learn about health,
Yeah.
about disease, just by looking at these protein patterns. Doesn't it make you wonder what other simple but powerful techniques might still be out there, just waiting to unlock the next big biological mystery?